Hello, I'm James. I'm going to talk to you today a little bit about neurocellular automata and how I've trained them to basically respond when you poke them. My clicker isn't working for some reason. There we go. Now, hopefully we all know about cellular automata, and I don't have to spend too much time explaining what they are. My view of them is that they have a state and they have an update rule, and that update rule historically has either been created by hand, by obvious people like John Conway, or we've trained some kind of genetic algorithm based on a fitness function, where that fitness function is describing some macroscopic property, and we're hoping it's going to come up with some update rule based off that macroscopic property, which gives rise to that emergent behavior. Now, neurocellular automata, as introduced by various people in this room, update that. I'm skipping over a lot of cellular automata research here, so don't kill me for that. Where instead of just having a binary state, they have a state vector of real valued numbers, where the sort of top 4 in that vector represent some visible state of red, green, and blue, and some alpha state, which describes whether something is alive or how alive that particular thing is. And then there are 12 hidden channels which aren't displayed visibly, but which are used for communication between these cells. And then we can do fun things like throw it at a neural network in order to produce an update rule. So instead of just handcrafting this update rule, we can now use, convolutional neural networks to train in some behavior which we can describe at that top macroscopic level now. It's all fairly well established by the gentleman at the back there. I won't dwell too much on that because I want to get to poking animals. That sounds weird. In sort of the early work, we could train multiple different high-level behaviors into these. When I started looking at this, I was like, this is really cool. We can build artificial creatures from a cellular level, from that kind of embryonic level, and grow them. That means we can study how things like cells differentiate in this artificial system, et cetera, the usual morphogenetic questions. The problem with this is that this requires training the network 10 different times. So we have 10 different neural networks, one for each of our creatures, which is growing here. So in order to realistically be able to compare how cells are differentiating in different ways, we ideally want them within the same network. That way we can trace individual cells as they're growing. So in my view, I've kind of, and don't kill me for using this particular description, I've kind of been looking at signals and the idea of grouping different signals as I see them, as either internal, external, or societal, where internal is typically anything coming from within an organism, genomic signals during development, hormonal signaling, et cetera, et cetera. Your external stuff is stuff coming from the environment. So anything like a chameleon reacting to camouflage, predators, that kind of stuff. And then some kind of societal stuff, which I've kind of described as stuff which is external to the individual, but maybe internal to a group or a society. Things like the same species interacting. Now, in reality, I'm more interested in societal, but in order to get there, we have to go through these steps of building internal and external before we can then worry about societal. So that's very much future work. So this is what we're going to be focusing on today, is what I call internal and external signals. So particularly relevant work to what I'm about to talk about, just to briefly cover it. This time last year were 2 talks which kind of inspired this approach. The first one is this adversarial takeover, where a new organism is implanted within an old organism, takes it over and replaces it. What this generally is focused on is you've got multiple neural network functions. How do they interact? You have some host, you have some adversary. How is one taking over the other one? Subtly different from what I'm interested in that I want it all within one network. And the other one is the environmental gradient sort of dynamic movement, where there's a consistent signal to the organism from the environment that it should go in a certain direction. The organism slowly crawls its way up that gradient or down that gradient or whatever. Now, to distinguish sort of what was going on here to what I'm going to talk about, I'm mostly focused on, can we get a single NCA to produce multiple organisms from the same network? So we're training multiple organisms into one NCA. And can we get an NCA to produce a single organism which can then respond to a transient signal? So instead of giving it that consistent gradient, we're just going to poke it for one time step and see what it does, see if it can respond. And these, roughly speaking, follow my internal and my external view of signals. So in order to do this, I basically borrowed a few of those hidden channels. So for internal training, this is basically encoding a genome into that seed cell. What I've done is I've borrowed 4 of these channels, these 12 hidden channels, and I'm going to start putting some data in there, which tells the neural network which particular organism I want it to grow into. Okay. The idea being that then when we trained it across multiple targets, we can then get it to grow whichever one we want by just changing this number. As an example, this is going to work. Not the best, but you get the gist. This is trained into a single neural network. All we're doing is changing channel 4 here to either be 0 or 1 in the seed cell. And then this is the same neural network growing into 2 distinct organisms, very similar. It's a very early result, but it gives you the idea of what we're really after here. What was more interesting when I started digging into it is how the neural network learns to generalize or interpolate some of these genomic signals, where, for example, in this example, I trained it with a small heart and I trained it with the big heart as a 0 and a 1. But then in testing, when I gave it 0.5, it was able to grow something which was halfway between the two. I also tried this with rotation. Didn't work so well because, as we know, NCAs don't like rotating very much. My animations really aren't coming through very well, so I apologize for that. I then wanted to see basically how far I could push this. How much could I fit into this one NCA? So I basically produced 6 different target images, 3 colors, 2 forms, and used sort of all 4 of our genomic channels to encode sort of ones if I wanted it red in C5, green one is C6, and C7 for blue, and then C4 tells it if I want a gecko or a heart. And as we can hopefully see, that will then grow into each of these shapes. But we can already see, if we start looking closely, a few more artifacts coming through. There's a few, starting to see the limits of, for example, our green gecko here, where the tail is starting to become blue. We're starting to see less clean form in the heart, where it's clearly struggling to distinguish between some of these at a finer level. Maybe it could have been dealt with another 15,000 training steps, but that wasn't really the point. So we can do some stuff. And the first question which I asked here was, is it only able to do these 6 forms? Because actually they're quite distinct. So maybe the attractors it's forming in space are quite far apart from each other. What if we pull them closer together? So I chopped some legs off a gecko. The idea being that these are then, very similar images to each other, where the body is identical on all of them, and it's just these finer legs which are distinguished. And then we're relying on 4 channels to just encode in each one of these legs. And what it's able to do is to grow all 16 of these quite happily. I've just knocked the batteries out the back of my clicker, so bear with me. Now, what was interesting of this was what happened when I had basically kept the damage functionality in, which if anyone's played with NCAs, is there as a way of helping that regeneration. And during this process, I realized that we could test what happens when we remove one of these legs from a grown organism. So what I was really interested in here was, how is it encoding what it is? Is it doing like we are doing, where we have a copy of the genome in every single cell? Or is it relying on that embodiment of the gecko with only one leg to know that it's a gecko with one leg? And the easiest way to test that is to grow one and lop off a leg and see if it grows it back. See how well this works. So we're growing a 4-leg variant, and we're going to chop off random legs. And for the most part, it can't grow it back. So what it's telling me is that actually it's relying on that embodiment of how many legs it has to know which attractor it's in. And what we're doing when we're chopping a leg off is we're basically giving it a kick out of that attractor, and it's finding whichever one it happens to be closest to, which makes sense, but not perfect. I'm going through this quite quickly because there's a lot of videos to get through, so bear with me if I'm rushing through some stuff. So that's internal signals. We'll come on to discussion towards the end. I want to get onto external signals because I think these are really cool. So for external signals, what I wanted was a way of providing a kick from the environment which the neural network can't write to. So I produced an extra layer in the state, sort of 17th channel down the bottom there, which is a read-only environment channel. This is basically fed into the neural network, but not in the update step. So the neural network can't write to it, it can only read from it. What this means is that I can then basically set that channel to 1 at any point, at any time, and it will feed something into the network. So the idea was that we were going to grow an organism to maturity and then give it this signal and change its target image accordingly to see if it can learn to distinguish or learn to associate, that signal with a change in behavior or a change in property. Well, we're just going to change the color of it for now. So after growing the heart, this is a really bad animation on Zoom, but there we go. We can see that if we give it the signal on the far top left there and on the bottom left, it grows into a green one initially, then it will turn to red with that signal. Then we give it the signal again and it turns back to green. If we just annotate that to make it a little clearer what's going on in the image, basically this is just me stopping the animation at different time steps to demonstrate how it's going through and how it's spreading across and responding at that kind of cellular level. And this isn't just a one-trick heart. It also works with geckos, and also a slightly nicer one for anyone like me who's red-green colorblind. Again, not the best video, but feel free to come and find me if you want to watch the videos on my laptop instead. And we can see that this is able to then do something similar. It's able to change color and then change color back again. And again, these are the similar kind of time steps. The stars are where I'm poking it. But then a reasonable question to ask, and fortunately the question I did ask, is how do we know that this green gecko down here is the same as this green gecko up here? They look the same. The loss function says they're the same, but the loss function is only focused on that red, green, blue channel. What it turns out is that they were different organisms, but there was no way of the NCA distinguishing them. So what happened was you could turn it blue, you gave it the signal again, it turned back to green, but then it wouldn't respond again. So I had to adapt the training mechanism a little bit to give it more than just 1 or 2 signals, to tell it that it had to go back to that original green gecko. So I did that up to about, I think, 10 pokes in training. I don't know how well this video is going to work because Zoom. And then to test it, I just set it going for a really long time, like 10,000 time steps. It grows in about 100. Then I give it a signal every 100 time steps. So there's roughly 100 signals being presented to it. It'll fast forward in a second. You don't have to sit here for 15 minutes just watching this swap backwards and forwards between 2 colors. However, what that demonstrates is that actually that small shift in what we're doing at training, where instead of just relying on, okay, change color, okay, you've done that once, change back again, that wasn't enough. What we needed to do was make sure it was going back to the original form which we originally designed it to do so that it can continually do this. And this goes on for quite a long time, doing what it does. Somehow I'm on time. As any of my students will tell you, that's rare. So in conclusion, we can train NCAs to respond to signals. There are various different forms of signal. These 2 types of signal we have managed to train into it. We can train multiple organisms into one NCA, distinguished by this sort of genomic signal which we provide in the seed cell. And we can grow organisms which can repeatedly respond to environment signals. So we've done kind of the hard work. Now we get to do the fun work, which is, can we now that we have the ability for these to respond to signals and we know that they can we get organisms to interact? Can we grow multiple geckos in an environment and have them interact in some way and train them to be able to do things, who knows what, fun things. The other thing which I'm interested in, which I started working on, but then I didn't include because NCAs are buggers sometimes, is can we produce movement from a signal? So originally I was training a gecko tail to curl one way or the other, depending on where I poked it, with the idea of going towards sort of muscle innervation. But if you poke it on one side, it curls towards that side. The other side, it curls towards the other side. Problem is, cellular automata being cellular automata, it just chops off the tail and grows a new one, which isn't very useful. And the other thing which I'm working on, which I have some results for, but I'm not 100% in them, is some information theoretic analysis of this to show how these cells are responding to that signal. The numbers look ridiculous and I don't trust them, which is why they're not in the paper or the presentation. But if anyone's good at information theory, come find me afterwards. For some reason I'm missing a slide. Okay, doesn't matter. There we go. That's that.